hope it's going to be an interesting, exciting, and fun chat. So Jamal, thank you so much for being here with me. It's exciting. It's great to see you. I haven't seen your face in a little bit, but you look great.
Yeah, it's been a while, and thanks for having me. Just to put it out there, I am a few years older than Ben, but Ben was like my guy to like look up to. He was a year ahead of me, so I've been following in his footsteps. I'm even out here on the West Coast, a few hours away. So we never actually worked on too many projects together, but we always like had lots of discussions, right, and came up with super creative ideas and like put them into the practice, right?
Yeah, absolutely. Well, I appreciate that, Jamal. Thank you very much. But my mom is here. She said, "Hi, Benny. Hi, Jamal." So
Oh, your dad's here too, or?
I don't know. Yeah, looks like it. Yeah, he says, yeah.
Nice.
So let's go ahead and get right into it. So let's talk about genetics. Let's talk about neuroscience, and let's just chat. And by the way, and also let me say, yeah, so Jamal's out here starting his postdoc at UCLA now. So both of us, two fellas from cold Buffalo, New York, moved out to sunny California. But
Yeah.
I'm really excited for Jamal. The lab that he's in is absolutely incredible. Truly one of the top labs, arguably maybe the top lab in this field of research. So, and my dad said, "Congrats on your PhD, Dr. Williams." Awesome, absolutely, congratulations, man. I'm so excited for you. Let's talk about all this stuff. So, I think a lot of people generally understand that your genes are critical for determining what you look like. You know, that's where biology classes typically go, right? It's like you have a gene for red hair, or you have a gene for freckles, or something like that. But I think that what's not really taught is that your genetics ultimately determine how your brain functions, because your brain is all your brain cells are full of genes which all control specific functions. And if you have, let's say, a gene mutation in a certain gene that's critical for one function, it can completely throw off the way that your brain is functioning, and that can lead to changes in behavior, um, and it can lead to disease. And I just think centering on this idea will be critical for this casual conversation. But you are now working on studying this specifically in like a really high level advanced context in a really great lab. Um, so beyond this idea of like knowing that certain gene mutations can lead to disease, can you try to explain, it's kind of a tall task, but sort of explain like how scientists study this in general? You know what I mean?
Yeah. Well, as you said, we think of genetics in terms of like what we get from our mom, what we get from our dad, that make up 23 pairs of chromosomes. And we think about things like height and, like you said, hair color. But also, if we understand the fundamental concept of biology, that every cell that divides and during development, from a simple zygote, will have the same DNA. So if we understand that DNA is our instruction for how we create proteins, and which those proteins like create like who we are, then we also accept the notion that in every cell that divides, you're going to have the same framework. And so what we're talking today is mostly about genetics, right? But you almost can't talk about genetics without talking about a little bit of epigenetics, which is something that we kind of did our PhD in. And so we have a basic instruction in every cell, but our physiology like needs to tell the cell like what to be. Like we need a cell for hair, we need a cell for skin, we need a cell for brain. We need many different types of cells for brain cells, right? And so although there is this framework that's in every cell, there's also these like proteins that come in and change how those certain genes are expressed. And so, in cases of mutations, which we see in disease, especially like heavily more genetic orientated disorders such as autism, uh, that framework is is usually like pretty critical when the brain is forming. And we're starting to find out now in like the last 10 years that when we see very severe brain disorders, that's because there's a mutation that's super rare that's in every cell as your brain is forming. And now through research, we're starting to be able to try to pinpoint, right, like where exactly in the development of your brain that breakdown and the dysfunctional gene is occurring, and how we can go in either with gene therapy or we can go in with different therapeutics to try to put things back into their proper order to help with brain development and certain like brain dysfunctions. It's a very, very, very difficult task. And you asked how we study it as scientists. Um, I think one important thing to note is that as scientists, we are kind of applying classical experimentation to discover these genes and their biological implication. But I will say, like, being in this field, like the higher like we go, it's evident that we need more than just scientists on board to help us sift through this genetic information to kind of peel back what's going on and like how to correct it. So right now we need engineers, we need computer scientists, we need people from all different types of backgrounds to say, hey, I want to make a meaningful impact, on disease, and I think that can have a meaningful impact on society. And although I'm not a scientist, maybe I can apply my skill set to science and help these individuals who have more of a biology background deal with the large amounts of data we're starting to get in about genetic disease.
Well said, absolutely. And we also need volunteers. We need people to, provide, DNA samples to help us understand specific diseases. But yeah, I mean, genetic data is inherently huge data. In science, we call it big data a lot of the time.
Yes.
But in order to sequence someone's genome, I mean, it's 25,000 genes, somewhere around there. So there's a lot of information. There's a lot of genetic material and a lot of data to be sorted through. So yeah, absolutely, we do need engineers. And another thing you said that was super interesting, great point, is that genes are used differentially throughout life. So this fundamental idea that everyone should understand, and you must understand in order to like advance through this conversation, is that what you said, genes are a script, a blueprint for a protein. And I think that understanding, like that piece of knowledge, is lost on a lot of people. And that like your DNA isn't just like your DNA in that it helps you, like it's a code for your body to like grow. And now you're an adult and now you have like your body and your genes have been used and now they're just kind of silent. I think that's how a lot of people think about things because of that explanation that we get in high school and all that. Like I was talking about earlier, where you have a gene for red hair. Okay, so it's active when I'm in development, but now it's done, right? Yeah. But the truth is that genes are always there as a blueprint, but they're being used at different levels throughout life, and that can be changed. Part of that is pre-programmed, right? So like a gene might only be necessary for like defending against like aging. It might protect your cells from aging, and your brain cells don't need that protein when they're super young, when you're 4 or 5 years old, but it does need that protein when you're 85, 90 years old. And so if you have a mutation in that gene, you're not going to experience anything different until you reach 85, 90. And that's when you're going to start to see something like Alzheimer's. Whereas if it's a gene that's, critical for helping cells grow, right? Helping brain cells proliferate throughout development, that's going to be critical early in life and early in development. And you might see something like microcephaly, where the brain size is smaller because the cells aren't proliferating properly. So I feel like that is such a critical thing to understand. I'm really glad you brought it up. Do you have anything to comment on that?
Yeah, I have a quick comment on it, but I just wanted to ask a question. Does your audience have access to the chat?
Mm-hmm.
Can we get a couple people to put in the chat, like, so if we learn from basic biology that DNA is A, T, C's, and G's, we call those nucleotide or base pair. Can we take a guess, or maybe you might know like what the approximate answer is about how many base pairs are in the human genome? Like, just throw out any number. Like, don't be shy. Like, it's really something that may not be like super common knowledge, but it does help frame a lot of what we're talking about.
Yeah, that's a great question. So if one gene is made up of a sequence of individual, like you can picture the DNA molecule like twisting, and there's little ladder branches. Like one of those is a connection between nucleotides.
Yes.
How many of those do you think make up the entire genome, all of your genes?
You gave us maybe you might have given us a clue, or you might have given us sort of a distraction. You said that there's about 25,000 genes. So there's about 25,000 genes, and genes are made up of these base pair. About how many base pairs do you think are in the human genome? Do we have any answers?
So my dad said, and please feel free to be brave, throw an answer out. My dad might be privy to some knowledge. I'm not sure. His guess was 1.3 million base pairs. I actually don't know the answer to this. I'm really hoping that you do, Jamal, and that you didn't just bring this up. Yes. A lot.
Okay, 1.3 million. Okay. Somebody said a lot?
Someone said, no idea. Sorry. A lot. Definitely a lot.
Okay, well, I'll give you the answer, but I got another question. This question is important, and I hope you can engage and throw out an answer. It's about 3 billion base pairs in the human genome. And so the next question is about what percentage of that 3 billion do you think is actually, let's use this as a catch-all phrase, like functional, like how many of those base pairs, like what percentage do you think have like some directly functional relevant like role in human physiology?
Oh, I'm nervous about this one. I don't actually know. Again, I don't know the answer to this either.
So we got 3 billion base pair. Just throw out any percentage between 0 to 100 of what percentage of that genome is function. Like 50, 60, 10, 100.
So probably from the same. Okay, so we got 5 to 8%, 10%. That's what I personally, my naive thought would have been like, you gotta hope it's like 95. You hope. Like there's 3 billion base pairs. They better all do something.
All right. So who said 5 to 8%? Because
that's my dad again.
Man, your dad is smart. I think he's getting superpowers from that neuroscience shirt I designed that I seen him rocking, and I think he's.
Oh yeah.
So close, actually. The answer is approximated to be by leading scientists in genetics to be between 2 and 3%. Wow. So those 25,000 genes that Ben described earlier are made up of just about 2 to 3% of the genome, and the rest of it, like 98%, was for a long time thought of to be considered almost like dark matter. So like we have no idea like what it does. Like now we give it more of a general term. We call it regulatory elements, but we still don't actually really know what that means. But the reason why we have to go through this kind of process just now is to understand that we're talking about mutations. And if we pick a disorder like autism, maybe we found 1 to 200 super important like mutations in genes. So like 1 to 200 genes maybe is affected, 200 at the very, very top level so far out of like 25,000. But actually most of the mutations that we see in our genome come from that 98%. And so we have some big data on our hands to sift through. And this is why I suggested that if you want to make a change, want to make a difference in like how we approach disease, and you bring any type of background, it could be graphic design, it could be, computer science, like we need all hands on deck to deal with this 3 billion base pairs that we only really understand about 2 or 3% about.
That's like borderline unfathomable. Like to think about all of that. And what's, if we know anything about biology, it's that the way cells work, the way the brain works, it's all purposeful. Things don't happen unless they are, or at one point were useful, because biology is like cutthroat. If it doesn't work, chop it off. We don't need it anymore.
Yep.
And if there's so much DNA that is just doing like we don't know what, there's something there. That's going to be so exciting and interesting when that becomes a well-known thing. Like that's a Nobel Prize for sure. Whoever figures out what the hell's going on there.
Yeah, and do those mutations actually mean anything? If you're just mutating an area of the genome that we can't really identify as having like a functional role in any sort of gene, then does that even matter? We think it does, but we're not entirely sure. So I guess, if you've been watching Ben for long enough, you know that we as scientists, the more we learn, the more we know the more we understand we don't actually know. Like the more questions we kind of come up with, and so this is a huge question and a topic of today's talk.
Yeah. Any guesses? Any theories on what it's for?
No.
You don't have any.
uh, yeah, well, we think that it actually plays some type of so when we think of DNA, right, we usually think of this two-dimensional picture, like you said, a ladder, but it's actually almost like a ball of yarn. And because if you have 3 billion pairs, uh, just those, the 3 billion of these like compounds, of these things we call like nucleotides, like where are we going to fit all of that stuff in every cell? I mean, how many cells we have in a body? Like, I have no idea.
Not a clue.
It could be. It's been estimated to be like 10 or 100 trillion cells. So it's like, how do we put 3 billion base pairs into trillions of cells, right? So it's condensed into something called chromatin, which is like a ball of yarn. And so that 98% of the genome that we really don't understand could be involved in some kind of three-dimensional functionality in terms of like allowing certain genes to be turned on or to be turned off. And Ben mentioned something about aging, like, throughout the aging process, you start to have certain genes turn on or off that wasn't turned on or off when you were younger. And um, that kind of like breakdown is based on how open or closed the DNA is to these guys who come in and turn a gene on or turn a gene off. And so maybe that kind of dark matter almost is like a part of the regulatory process that allows this ball of yarn to be a little loose or a little tight. Sorry, I'm next to the hospital, so it's lots of helicopters every day.
Yeah, people getting airlifted in all the time.
Yeah, and it's the only place outside. Is the only place in this entire university I can find where I can talk to you guys without wearing a mask.
Well,
I'm not that pretty to look at, but it makes for a better conversation if you can see me.
Yeah, no, we appreciate you being here. No worry, the helicopter's not that loud. I can hear it, but it's not overwhelmingly loud. But yeah, so that's what. So like you were describing, regulatory elements, right? They're some way they allow genes to be regulated. What my dad commented is maybe they're learned defenses against other threats. So there's also this question of like, I think about this all the time. A deer in the wild, it knows not to eat certain berries because those berries are poisonous, let's say. It knows to eat other berries. Do you think that information, that sort of instinctual information, is contained in the genome, or like is it contained? Is it learned? Is it taught? Uh, I always think about this. There must be a way for information to be genetically inherited, and epigenetics seems like a plausible route for that, a plausible vector. I don't know. It's so intriguing but complicated.
Yeah, it is pretty interesting to think about. I would say yes, it has to be inherited for sure, because there's examples that I've come across, like just reading books by like Richard Dawkins, and he's like a famous ecologist, and learning about like different types of birds who have like this like instinctive like survival like mechanisms, and you have to think that that's inherited because there's actually no chance for it to be learned. And so, in some different species, the offspring is left by themselves to grow and adapt, and they grow and adapt in exactly the way that they're supposed to do. And so how do they learn that? Even in terms of like um, I don't know, maybe that's not a good example, but I mean, even as like humans, for example, we don't actually, like mothers may take a class on like how to be more effective at breastfeeding, but it's like, how do babies like know like where to get their milk, and like from day one, like a
Right.
baby is born and it's immediately given to the mother and then it immediately starts to be able to eat. Like, how is that learned when the brain is so small, when there was no training, when it's never seen this act done before, but the baby knows this has to be done for survival?
Yeah, absolutely. I've never thought about that example before, but that's an incredible one. Like, it's unbelievable, and it's such a mystery, and I find that to be so interesting. I find a lot of different things to be interesting. That in particular is like towards the top. But I actually have thought about doing an experiment like this. And I talked about this on another podcast that I was on, and now I feel comfortable just talking about it freely. But this idea of like taking a mouse, let's say, and exposing it to another mouse. Okay, sorry. Taking a mouse, exposing it to like peppermint, but also giving that mouse like something to make it feel sick. Then exposing another mouse to that mouse. And would the other mouse avoid peppermint if the mouse smelled like peppermint and was sick? And I would argue that it would definitely avoid peppermint after that. There is evidence for social transfer of food preference, so in the opposite direction. So I would assume it would work in the negative direction. But then my question would be, would the offspring of the mouse that witnessed the peppermint mouse, would its offspring also avoid peppermint? And if so, now you have a model to be able to study how these learned experiential understandings are inherited. And I would argue that there's some sort of epigenetic marker that can be detected, but it would be so challenging. It sounds straightforward, kind of, but it's where would you know? How would you know where to look? What would you look for? But
Yeah, I think that would be a super interesting experiment, and you're right that it would be super complicated because we don't know what genes would be involved and where those genes would be at and in which areas of the brain. But in a sense, it has to be. And like we kind of explained what epigenetics was at the beginning, it's just, a system that allows certain genes to be turned on or turned off, genes that already exist in the genome but are either expressed or not expressed. But epigenetic changes can be inherited. Actually, some of the first epigenetic studies looked at inherited changes. So something that happened during a person's life being able to be passed down to their offspring. Some of it was trauma. Some of it was early studies was malnutrition, and something like fear could be a mechanism that is like passed down through epigenetic changes.
yeah, it's such an interesting topic. Sorry, now there's like a huge like war plane flying by my house. Luckily, not that loud. So let's go back to this question of how scientists study genetics. What often happens is that if you're interested in studying like autism, for example, scientists may recruit a bunch of study participants who have autism spectrum disorder and obtain some DNA sample and sequence it and look for specific mutations in various genes. And what might happen is that they see out of 10,000 people with autism, 400, or no, that's pretty high, like maybe like 100 of them all have this same gene that's affected by a mutation. And these studies are called GWAS, genome-wide association studies. And they're very prevalent. They're growing. I've noticed there are some like criticisms of them, I suppose, but they're very powerful tools to at least like give some sort of indication of what genetic elements are affected in specific disorders, especially if you can identify that in this group of 10,000, maybe gene mutations were only identified in 4,000 of them. But of those 4,000, it wasn't that there were 4,000 gene mutations. There were 40 gene mutations. And maybe all those genes are related to the same types of processes in cells, which can help us understand that biological process may be relevant to that disorder, whatever it is that we're studying. And so this is a really powerful tool. Wanted to ask you, Jamal, so like how do you view GWAS studies? Do you have any thoughts on them? Do you think they're powerful? Do you think they're they're limited. Like, what do you think is the utility of these types of studies?
Yeah, I think it's super powerful in terms of being able to identify um, what we call like single nucleotide polymorphism. So changes in one of those base pairs or one of the letters that make up the base pair. Uh, but in order to conduct this, you need a lot of people, right? If you want to just have statistical power and find things that are changed one nucleotide at a time, you need lots of people. And then also, if you're able to find changes that are significant, by definition, they almost have to be common in a certain group of the disease model. And so we actually call those like common variants. And common just means like it's in like more than like 1%. One challenge to, so it's very powerful. One challenge is that the studies may not be that effective for people who suffer from certain genetic disorders. Let's just talk about brain disorders that have what we call like a large effect. Because if something is common through evolution and natural selection, we typically would have rooted out something that wouldn't allow us to have enough fitness in order to thrive and to reproduce. So where we see a lot of like high effect in brain disorders is in rare mutations, and those rare mutations can't really be detected so much by GWAS studies. They're more so detected by like whole exome sequencing, which is focused on that 2 or 3% that we talked about earlier, or whole genome sequencing, which is starting now to get the computing power to do, which is focused on 100%, including those 98%. And then we can kind of pick out different rare mutations, either at the single letter, at the, at 10 letters, or up to a megabase, like a million letters that are changed. Ben studied a lot of this during his PhD, where there's like a whole section where there's about 20 or 30 genes that's changed, because that whole section can be deleted from the genome completely. And so in every cell, you lose all those genes because you've lost all that genetic material, or that section is duplicated. Those we call copy number variations. And so going back to like the point I was talking about of the high effect sizes, those usually happen when the mutation is rare. And so GWAS can't really give us enough power to look at rare mutations, which are the ones that kind of clinicians are more like inclined to try to address to treat patients. And these rare mutations, they can be inherited, right? So you can have a rare mutation that you get from your parents, but they also can be random, right? And that's when it really gets scary. And so those rare mutations can happen in the sperm, it can happen in the egg, or it can happen in the zygote. And so the child can have a mutation that leads to a very high effect from a very rare mutation for a certain disorder that neither of their parents have, and maybe, and most likely, none of their siblings would even have. And so if you experience something like that, that's where the disease and disorders have the really like highest like effect.
Yeah, and that's a great point too. I think parents who have a child with some gene mutation, whatever, affecting any gene, I think there's often this confusion or this question of like, what happened? Maybe some parents maybe feel like a sense of guilt, like what did I do? And it's just completely random. These things can just occur randomly. Um, there's essentially no prevention. But one thing that I want to touch on, which you mentioned, is that you said if anything above 1% is common. And I
Yes.
think to anyone hearing that, they might think that makes no sense. Anything above 1% is common. That sounds so silly,
Yeah.
but it's the case. I mean, for something like autism, there are, like you said, maybe 100, 200, maybe more. Who knows? We haven't yet really. We kind of have explored a lot of
We
that,
haven't found all of them yet.
but we probably, at least 100 to 200 that we know are like pretty strong for predisposing for autism. So this is what we call polygenic. It's many genes that can predispose to a specific disorder.
Yes.
But something like Huntington's disease is monogenic. So there's literally a gene, the Huntington gene, that if you have a gene mutation in that gene, then you will most likely have Huntington, or almost certainly have Huntington's disease. So for a gene to be above 1% in an entire population, it may sound like a small amount, but it's actually pretty strong if it's a polygenic disorder. So that these are all the things about genetics and neuroscience that you don't really understand or hear about until you actually dive into the research and start, studying it yourself. So
Yeah, and you mentioned something about parents, maybe there being like some form of guilt, but every time a cell divides, every time DNA is replicated, you have mutations. It's just going to happen. Um, Most of the time, it doesn't matter. It doesn't affect the proteins that are made, and it doesn't affect. We have no real like visible phenotype for this. Even the de novo mutations, we get de novo mutations. Like every offspring has de novo mutation, but it's when it's so unlucky that the mutation happens to fall within the gene that causes a protein to change within a um, just this certain like identifiable factor. Like for example, going back to autism, because out of all the brain disorders, it's the most studied in terms of genetics. Most of the time, if you the 1 to 200 genes we talked about that are affected with these random rare mutations, they're related to what? Pretty much two categories. They're related to how neurons communicate with each other, so like what we call synaptic function, or they're related to epigenetics, like we talked about earlier, like how genes, like those guys I told you about that go in and change whether a gene is on or off, those are disrupted, and so we have what we call dysregulation in gene expression. And so it just so happens that it's so unlucky that you have a mutation that happens to land in a certain region that expresses for a certain gene, mainly related to those two categories. And that's where we see a lot of the problems occur.
Yeah, right on. I mean, I don't know the actual statistics on this, but I would assume probably every single person alive has a gene mutation, or multiple, probably many, probably a tons. I mean, a gene mutation is really just, if you have a gene, it's a sequence. It's like writing out a paragraph of text. And then if you swap one letter in that paragraph of text, and let's say, if I'm reading my dad's comment, any theories on what that remaining DNA is there for? Maybe learn defenses against other threats. If I changed the D in DNA to R to make it RNA, that would completely change the context and the meaning of that sentence. But if I were to change it to any theories and I changed it so that it was spelled Theeries, so it's just a misspelling, I still understand what he's trying to say. I still get the concept of that word. That's essentially what's happening. Is you can have a gene mutation where it affects this part of the gene that doesn't affect the function of the gene. Or you can have a gene mutation that affects the function in a way that it completely makes it dysfunctional. And similarly, you can have, out of a whole spectrum of genes, your entire genome, you can have gene mutations that cause serious problems and others that cause like no change at all. So, and it's also funny to think about. Like I was talking about this in a TikTok video, and someone commented and was like, if a gene gets mutated, but it doesn't affect the function, is it still just like a gene, or is it even like a gene mutation? And I was thinking about it, I'm like, no, it's a mutation. But if I have a gene mutation that's harmless, what's the word? Isn't there non-deleterious? Is that like a better
Yeah.
word?
Yeah. Uh-huh.
So a mutation that doesn't affect the function of the gene. And I
Or it
have,
makes sense. Yeah.
yeah, and I pass that gene to my children, and they don't have a mutation, or they have the same mutation but it doesn't affect it. Is it really like a mutation, or now is that just like the accepted gene body? It's kind of funny. And like the way we know what these gene sequences are is that we obtain DNA from people and we sequence it, and we look at what the gene sequence is, and that gene sequence that we consider like the standard sequence could have a mutation in there that's not problematic. So it's kind of interesting. And then down the road, someone, an offspring of mine or my grandchildren or great grandkids could have a mutation that does change the function of that gene and cause problems. So it's just really an interesting topic. I hope you can still see me. My monitor just went off.
Yeah, we can see you. Yeah,
Okay.
that's exactly right. But even though that's exactly right, it's more common that the mutations are happening in areas that's not genes. So we're always getting these mutations anyway, but they're in that 98% of the genome. But I guess it would be correct if it happened to somehow land within that 2 to 3% and it doesn't cause any significant change and that gene is propagated, then that is the new gene for you. And and that's just how it is. If there was a mutation that was pretty bad and that caused your offspring not to live past 12, then most likely that gene wouldn't propagate anymore, right?
Right.
Because it would be fended out by natural selection.
I'm so glad you said that, because that brings me to a topic which I wasn't planning to talk about, but I really want to. I think it's so fascinating, is the idea that gene mutations for something like autism are highly prevalent and highly common, because having autism doesn't affect your ability to procreate. You are perfectly capable of having children and living an entirely normal, healthy life. Something like Alzheimer's, on the other hand, it will slowly lead to, it will be fatal. It's a fatal condition.
Mhm.
But it doesn't happen until later in life, where if you have a gene mutation that causes Alzheimer's, you will pass it on, because it also doesn't affect your ability to procreate until when you pass the part of life where you procreate.
That's
And then on
right.
the other hand, like if you have like childhood cancer, like serious pediatric cancer, gene mutations for that are, I'm assuming, less likely, because they're not propagating within the population. Because unfortunately, obviously, some serious cancer early in life that ends your life will interfere with the ability to procreate. So I think it's so interesting how like evolutionary pressures will never select against predispositional genes for Alzheimer's and other conditions that affect you later in life. I think it's such a neat topic.
Yeah, I think so too. And it goes back to like the common versus rare and the effect size that I talked about. So if you see a person with a very severe form of autism that affects their IQ and their cognitive function, then most likely that happened from um, a rare mutation, and there's a good chance that that rare mutation was random and wasn't able to be selected against through heritability.
Mm-hmm.
So most likely those individuals probably won't like procreate, but most individuals with autism with common variants will have a normal life to be able to sort of do what they want, or and have kids. And so what's being passed down will probably not be a severe form of that disorder.
Mm-hmm. My dad, who is a computer programmer, commented, he said, thinking about that dark matter. As a programmer, I recognize that a computer's memory contains both programming code and data that works with the code. I wonder if that dark matter might actually be data that the active DNA uses as a template to configure those memories, like which berries to avoid.
Wow. Yeah. See, this is why we need all hands on deck,
There we go,
because
Dad.
that is the leading hypothesis right now. That something along the lines of what your dad said is amongst the leading hypothesis of what's actually going on.
You're hired, Dad. Jamal is taking you into his lab. Um, so, all this talk about genes and disease and all this stuff. Let's talk about how someone, and I'm not sure if you actually can answer this, if you know the answer to this, how someone can learn about their genetics. Like, are there sequencing available? Can you get a clinical sequence genomic sequencing done to identify your risk factors?
Yeah, so I think I'll start with the most important ones. Is when you suspect that your child may suffer from a certain genetic disorder. Mm, I can't speak too much on this, but from everything that I do know, I have the strong impression that it's very difficult to actually get your child's DNA sequenced unless you're a part of a study,
Mm-hmm.
because there's probably complications with insurance willing to pay, and that is a huge roadblock in like discovery in science, because we really could use that data. We could use the people who are willing to contribute their data to study disease, and if we were able to get those paid for, then we would have that information, and most importantly, we would be able to maybe um, more selectively target individuals for a certain disorder and screen them and diagnose them very early. And so most people by now have heard of precision medicine, which is exactly what it sounds like. It's like giving an individual kind of therapeutic approach to their particular disorder. But there's going to be a huge discrepancy if only the people who can afford to have their genome sequence are able to get this kind of template on a table in order for a clinician to say, like, this is the specific, precise therapeutic that's designed for you. For
Yeah.
everyday people who want to get their genome sequence, you could do like one of these companies such as like 23andMe, and um, that has to be a personal decision whether that's something you actually want to do, is give a company access to your data, and if you think the trade-off is worth it. I'm actually indifferent about it, so I can't really, I don't really fall on one side or the other, to be honest with you. But that's a personal decision, and you can find out things like, at least a few hundred genes that you might be a carrier for, or you can find out things about your ancestry. So yeah, there are things like that that you can get. It doesn't give you the whole, like, sequencing in terms of like looking for other types of disease, but there's about a few hundred pretty common ones that you can get screened for, and that's about $200, I think.
Yeah. Uh, Super random thought, but I just moved recently and I live right down the street from 23andMe headquarters. It's kind of funny. It's like right there.
Wow.
Um, Silicon Valley, I guess you see companies like everywhere. But you brought up two things there. One being that you can get screenings to look at your ancestry, and the other being that uh, in modern day, these clinical screenings, these clinical genetic screenings, there's a disparity in their availability based on socioeconomic status. And it leads me to a topic which is important: the historical exclusion of certain groups in these genetics types of studies. Like many things, uh, these studies have favored white European populations, and they've generally focused on those populations. But I mean, is there anything going on that is seeking to correct that? And what issues could that introduce? This focus on white European groups, what issues could that introduce to our current understanding of genetics?
Yeah, so I think one of the kind of classical figures that came out in the paper within like the last decade showed in there's a Nature write-up, um, uh, that there's about 96% of the genome that we understand is from European ancestry, and literally everybody else is within that 4%. So why is that a problem? Well, I mean, there's an obvious problem in the sense that maybe we don't understand so much about the disease, right? If we only looking at one type of population. Um, and there's another problem in that, again, when we talk about precision medicine, that um, where we only have enough data to understand ancestry from people with European ancestry, and people who make up the majority of the world are all fall actually within the smallest percentage of that. So we can do like a thought experiment, right? So when we do like certain analysis in the lab, we need to map our, let's say, gene expression data to a genome, right? So like when you ran RNA sequencing for one of your projects, which is where we measured the gene expression for every gene in the genome for a certain organism and compared it to, one with a disease, one without a disease. We need to map those fragments of DNA to a genome build, right? We need to know like where they fall. That was the whole point of the Human Genome Project. But let's say that the map that we have is only represented by European ancestry. So from the very beginning of our analysis, it's already slightly flawed, right? Because we're mapping the gene expression for individuals who may not fall within that ancestral category to a build that is mostly the same, 98% the same. Another 98, not the first 98% I talked about, but just the similarity, like 98, 99% the same. But that 1 or 2% difference, or less than 1%, that 0.5% difference could matter if we're talking about something as direct as precision medicine. Mm-hmm. So our lab right now is leading a kind of collaborative nationwide project to specifically target African Americans with autism and offer them services such as diagnoses and early intervention, and get those patients who, or in families who volunteer to give their data and sequence those and look for maybe different mutations that and risk factors that exist within those populations, but then go back and figure out, well, how do the kind of characteristic that's caused by these mutations converge with what we already know about a disease. So we're not saying necessarily that the disease has to be different. It can be, but what we're saying is that the risk factors, the diagnostic criteria, if we're going to scale up this precision medicine, is completely missing large groups of people. And so if we have that data, now we can figure out what those risk factors are and kind of what categories about the disease do they that we already know do they fall into.
Absolutely. Well said. I mean, like you said, a 0.5% difference, you might say, oh, who cares, 0.5% difference. But if someone has a SNP, a single nucleotide polymorphism, a single point mutation in a gene, and that gene is Huntington, and they have Huntington's disease, that is a like 0.00000 times 10 to the 50th 1% change in the genome. And if we used that genome as our model genome, so let's say like as our white European genome, we would be basing all of our studies off of a person that has Huntington's, which is a very serious condition. So it doesn't make any sense. It's difficult to, sort of like homogenize all populations into like a standard, this is what the genome is, but it's also extremely important to at least understand the differences across various ethnic groups. So, I'm really glad to hear that that the lab is working on that effort. I think it's fantastic, and I'm excited to hear it. I'm excited to hear about it, and I will. I know I will. Yeah. So, I was, let's go back and talk about this precision medicine idea. So I will share with the audience, like I said, Jamal and I have known each other for a while, all through our PhDs, and Jamal is an absolute visionary. His vision is unbelievable. I will say there are vaccines being developed nowadays for Alzheimer's disease, and Jamal had the idea first. He did. He told me about it. And I swear to God. But yeah, Jamal has always had these amazing ideas. Speaking with you has always been fantastic and extremely insightful, and helped me think about things in new ways. So I want to ask you two things that you've shared with me that I think you should share with the world. One being your vision for the future of precision medicine and like patient-tailored care. And I'll get to the second one after. So if you're comfortable talking about some of the thoughts you have on that topic in general and where it could go in the future.
Yeah, I think first of all, we need to establish trust in communities that we've excluded, because not only is it the right thing to do, but it's imperative to understand disease in its entirety. You cannot just take a small slice of the population and use that as, the dominant um, background for a particular disease or for any disease. So what I hope for in precision medicine is that we can get everybody on board in a meaningful way and really understand disease as it affects different populations based on, geolocation, based on ancestry. Um, we really need this information if we are going to, um, build this foundation for precision medicine. And the second thing I hope is that um, the availability of precision medicine becomes more equitable. And so Ben talked about me being a visionary. I have lots of visions for sure. I have lots of ideas that, admittedly, I do think are good ideas, but I don't have the technical acumen to do a lot of those things. And so, as a part of this kind of scaling process and dealing with big data, I have to say for the third time, I really hope we get people from all types of backgrounds to jump in and say, I'm ready to do something about it. Because while I can use my kind of societal and biological background to think of good ideas, it's no way possible I can develop the necessary skills to do everything that I feel is needed to be done to achieve these. And I don't think people like me can do all of those things. But hopefully we can collaborate and make those things happen, which is why when I build my own lab, I have a super lofty idea of what to do. And maybe that's what Ben is talking about, something that I discussed with him before.
Yeah, you don't have to share your long visions, your goals, but yeah, and just in case anyone's unclear on what exactly we mean by precision medicine, it's just the idea of like if you have if you're diagnosed with major depressive disorder, typically the immediate response would be, okay, you would like to try medication. Well, we're going to give you an SSRI. Which one? I don't know. Let's try this one. It doesn't work. Okay, well, now we have to switch you to a different SSRI. Let's try a few different things on and on until you eventually settle on a medication that might work for you, might not. Precision medicine is the idea of integrating your genetic background. This is one example of it. You're integrating your genetic background with your symptomology to help understand, like, first off, what biological process could specifically be driving these symptoms that you're experiencing, and secondly, what specific biological approach could be beneficial for you. So maybe we know that the problem is not serotonin. This is really simplified, but let's say SSRIs, they target serotonin. Let's say you have depression, but it's found that you have a mutation in some gene that's related to dopamine. Maybe some sort of other drug that targets the dopamine system would actually be a more effective intervention strategy for you specifically. So this is the idea of precision medicine. It's tailoring your medical care to your what we understand about your genetic background. My monitor turned off again, so I'm going to fix that.
Well said.
Thank you. So the second thing that I wanted to talk to you about is this vision, this idea that you have. I've heard you say before, and I actually don't know, we haven't talked about this in a little while. I'm not sure if you still believe this, but I've heard you say that you believe Alzheimer's disease is sort of an acceleration or an intensification of the natural aging process in the brain. First, do you still believe that? Do you have any thoughts about that?
Yeah, I still believe that.
So, well, my question is, sorry, I'm over here fiddling with my cord. What my question is if you're comfortable sharing, you don't have to be. This might be something deep that you're planning on studying down the road. But what evidence you saw that led you to that understanding of Alzheimer's disease?
Yeah, so first I would like to just say that, I don't like to make like too many like predictions or speculations of things that I don't know. I do like to envision things that I would like to see change in a very optimistic way. And so let me just, say that this is definitely speculation based on information that I know from doing my research and through literature in Alzheimer's disease. So earlier we talked about epigenetics, and we both talked about like through aging, there is just a natural process that happens at the cellular level, at the DNA level, at the gene expression level specifically. And so aging is in itself an epigenetic factor. It is an environmental factor. One of your previous guests, Megan, is a really smart MD PhD student from Buffalo. She thinks that Alzheimer's is mostly environmental. And I've come a little closer to that line, environmental outside of aging. I've come a little closer to that line since the beginning of this idea. But I think that there is genetics that we don't really understand that underline the framework of what's actually happening at the environmental level. Because if DNA is our template, and as you mentioned earlier, if that template is slightly changed, but we don't actually even use that area of the template for most of our life until we age. And when we age, we have a loss of what we call heterochromatin. So that ball of yarn I described becomes more loose, and genes that are usually off start to become on. So if the gene that if the protein that's made that sits on top of the gene that is made in the foundation from this like mutation that has been sitting there forever but was inactive becomes active now, then those proteins that ends up being developed may be slightly different, right? And so, yeah, obviously through aging, there's a protective process. And I don't know, through evolution, I think if it was just left up to evolution and we were any other species, we probably would still be dying between 30 and 50 years old. And so aging is kind of a sort of interesting environmental factor in the sense that it's new to humans. That being old is actually new. Just maybe 5 or 6 decades ago, the human lifespan was about 50 years old, 55 years old on average. Now it's about 77 in North America. 100 years before that, it was probably like 30s to 40s or something like that. In 1800s, 1700s, 1600s, you hear about so many of these like great mathematicians and physicists and these people we learn about from history that died, at like 30, 40 years old. So my point is, I still do believe that there's a genetic underpinning that we just don't really understand yet. That's probably happening in that 98% of the genome. Also in that 2 to 3%. And I think that aging and other environmental factors affect the way that those genes are turned on or off.
That is so damn interesting. I didn't know that there was a loss of heterochromatin. So I'm just going to reiterate this in case anyone didn't fully get this. Your DNA is packaged tightly in your cells. Like you were saying earlier, it's like balls of yarn because if it wasn't, it would be everywhere. It'd be gigantic. So it's tightly packaged. So as you age, the cell's ability to package the DNA tightly is lost. Is it a problem with like do we understand why, or is it just like it's seen less often, heterochromatin?
Maybe other people understand why, aging scientists. I'm not sure, but it's detected through methylation levels, which are major drivers of heterochromatin stability or
Wow.
instability.
So without getting too deep into the weeds, the idea that your DNA becomes more loose in the way it's packaged, which allows for more and more genes to be readily accessible, and therefore they are used more, they are expressed more, can lead to negative health, cellular health. It's really interesting. I mean, it makes sense. Of course, not all genes are good. You don't want all your genes to be expressed all the time. Some may lead to cancer, because if you have a gene that causes cells to grow and proliferate and it's being expressed all the time, that's something that's going to lead to cancer, for example. But I never thought about this idea that could be a natural process that is actually not helpful at all because, like you said, aging is new, it's unexpected. I actually just took like a whole journey in my mind while you were talking, and I was picturing like old people in my head and thinking about how like 200 years ago, like that would be such a terrifying sight because the old people like that didn't exist. Um,
Mm-hmm.
but that is so fascinating. So, do you have any intention of potentially exploring that hypothesis in the long run?
No, I don't. But I think now a lot of aging scientists are actually starting to figure out what they call put their effort into what they call health span, because of aging being such a new concept for humans. So again, it's our medical advancements and our ability to create vaccines, to create antibiotics, and our medical advancements is the main reason why we live almost twice as long as our ancestors. I mean, it's nothing else really that we did except advanced medicine, and it really accelerated to the point where we kind of, accelerated what would happen to us evolutionarily. I mean, it's certainly not that we created a better Earth for our species, and that's why we live longer. I mean, we all know that's not true. We destroyed this place, but it's through our medical advancements that we live longer, and those medical advancements are just a couple hundred years old. At the oldest, right? So now leading scientists are starting to figure out, well, okay, now that we're aging, let's understand this process a little bit better, and what this means, and what negative consequences of aging looks like, and how we can not only help people to like live longer or live past aging related disorders, but how we can also allow them to live like healthier, or kind of age in a negative way more slowly as you increase lifespan.
Yeah.
It can be done for sure. I don't like to make predictions, but if you can understand these mechanisms and how aging, this new concept work, you can definitely slow it down, because it's really all epigenetic.
Wow. Maybe there's a future. One of the other events I did on here was about bioengineering, and about how we can like 3D print organs and stuff. Or we can't, sort of can, but we're getting there. And it would be such a fascinating conversation to combine what you're talking about with that, or maybe in the future, if we can slow down the aging process, and then just like 3D bioprint new skin and new organs to replace like the old dead ones, dying ones. Maybe that's our path to living forever. Not that that would be necessarily good, but that would be so interesting to see, because there's this obvious change in the way the brain works, the way we think as we age. When you're young and this happens to me a lot, where I'll be like in a public place and I'll smell like a perfume or something like that, and it will immediately trigger this like deep, like really robust memory of when I was young and I was in like the library with my mom, and like a lady walked by with that perfume, and like for a second I can just like see the library and like, feel the excitement of like being there in that moment. And it's funny how life is not that way anymore. I don't experience life in that with that heightened vigilance and excitement and intensity. And I wonder like how just sad and monotonous life would become if we could actually live forever. Like, how would the brain interpret information after being alive for 200 years? It would probably just be like, I'm not processing anything right now.
Yeah, it could be. Also, again, to think about our planet and overpopulation. If we already did such a messed up job on taking care of the planet that we currently live in, what happens if we make more of us that we assume we're the most important species? What does your audience think? I mean, do people think that we should use science to advance how long we live? And if so, what's a good number that you think would be a cutoff where we can say, okay, let's shut the science down now. We can live to X age. Is there any thoughts about this?
That's a great question. I'll wait to see. You've also just made me. I like thinking about like sci-fi fantasies and stuff. But like, what if we did collectively as a society decide that like 150 years was enough, and then like at 150 you just get like terminated. That's it. It's like no more. Yeah, someone put a vampire emoji in the chat. Eternal life. Miriam said, I'm happy with 85 years. 85 sounds pretty good.
I'd be super happy for 85 years at this point.
Let's see. tough question. I think the degenerative processes would have to slow down. This sounds funny, but if I feel like this at 58, what would my body be like at 200? Yeah. I feel the same way at my age, and I'm not quite 58 nearly, especially considering that's my father saying that, so we can assume how much younger I am. Miriam said, don't ask me how I came up with that. 85? I would be sad if they decided to terminate everyone at 85. That was the cutoff. That seems a little too young. But yeah, it's an interesting question. I mean, these are the types of debates, hopefully not debates, more discussions that will become more and more feasible and realistic in the coming years. Also things like, like CRISPR gene modification. If you have a child and there's a gene mutation, how do you decide whether or not that child is eligible for some genetic manipulation? And just these types of ethical discussions, which will be happening at the governmental level. I really hope that scientists are participating in those discussions.
Same here.
Yeah, I mean, maybe not exclusively scientists. Ideally, a diverse representative group, but definitely not exclusively politicians. That would be unfortunate. But
truly.
Okay, so very interesting question. I think if we were able to live to 150, we would stop taking care of our health. And what Ben said happens to me all the time. I smell or a taste will trigger a memory. Yeah, I agree. I think at 150, we definitely would stop taking care of ourselves, especially if we knew that we had the ability to just like replace an organ with a 3D bioprinted organ if it shuts down.
Mm-hmm.
It's a dangerous game to when you start playing in those roles. But
I do think aging scientists have the ability and will have the ability within the next couple of decades to really decode the aging process. And so although we would still grow in years of age, I do think they will be able to figure out a way to slow down the biological aspects of aging. However, one of your astute audience members mentioned that we would probably stop taking care of ourselves at 150. I mean, in this country, we don't take care of ourselves in general at all. So even if we crack the code on aging and they said, hey, if you follow these like 10 rules, you could live to 200. I think, but one of those rules was like, you can't eat like a cheeseburger, or, you have to move around like 10 hours out of the day or something. Like people would be like, yeah, whatever. Like, I don't need 200. Right. So I think much, much earlier, people would probably just opt out of like doing that and just like say, hey, I'd rather have like an enjoyable life and eat fatty food and be lazy a little bit sometimes.
Absolutely. That's so true. I mean, we already know that like exercise, diet, sleep, these things are all great for, extending longevity, but still people are like, no, I'll skip the gym. I'll take that cheeseburger. I'll, stay up all night. It's kind of funny.
For sure, man. I'm going to walk to Chick-fil-A when we get off of here.
At least you walk.
Yeah, I'll tell myself that,
Now you got to drive. You're going to call it.
I'm not going to get my car, and drive over one of those power scooters out here in LA and go get a Chick-fil-A sandwich.
Yeah, it's the great issue, right? It's so hard to practice what we preach, but at least we can preach it, and maybe someone will listen and say, all right, yeah, I'll exercise. I was just giving a talk right before this, and that was my last slide, is like how to maintain your brain health. And it's just exercise, diet, sleep, exercising your brain, working out your mind with cognitive exercises. But like, I don't do half of that stuff. I know it works, but I need to be more aware of that, more present. But anyways, I really enjoyed this conversation. It's been great to see you again and chat with you again. And we'll definitely have to meet up face to face sometime soon. Either I'll make a trip down there, or you can come visit me up here in the Bay Area. But
sure.
Yeah, I really appreciate you taking the time to join me here and talk about all this stuff. And I hope it was pleasant and educational for people. And I hope it made people think, most of all. That's my goal, is to just sort of expand people's perspectives and offer opportunities to think outside of the box that is your own mind. So thanks again, Jamal. And yeah, I'll see you next time on the show.
All right. Thanks for having me.
Of course.
Take care.